Dear students, welcome to Bioratna. I am Chandana Sengupta and our today's topic of discussion is mutation. Mutation, it is the change or alteration in DNA structure. So what is mutation? It is the alteration in DNA structure. So naturally it will cause the change in genotype of the organism, which will subsequently cause the change in the phenotype of the organism. Change in phenotype. There are two factors which are greatly responsible for this alteration or change in DNA structure. What are they? One is recombination. This recombination, it occurs during prophase 1 of meiosis, and another is the one which today we are going to see, that is mutation. See, this one chromosome I am drawing. So this is one chromatid and this one another chromatid, and in each chromatid the DNA helix, it is present in a supercoiled form. I repeat, in each chromatid DNA is present in supercoiled form. So if there is any deletion, or duplication in DNA, so naturally it will cause change in the chromosome structure, and this is called chromosomal aberration. Chromosomal aberration. Let us see what is deletion and duplication. Deletion. Fine. So let us take one chromosome and let us show the genes present in it. A simple way to do that, suppose you write A, B, C, D, E, then some dot. This dot is representing the centromere. Then E, F, G, H. You can write 2-3 more. That is up to you. But this much is sufficient. Now what you do? Now you draw this chromosome structure and then you show this gene on it. Done? Now in deletion what will happen? Any gene, suppose A or B or C or D or BC or DE, it gets deleted. When it gets deleted, when there is a deletion, then what will happen? Suppose the C and D has been deleted. So what you will get? A, B, E, F, G, H. So it is A, B, C, D is deleted. Then E, put one dot, and F, G, H. F, G, H. Draw the chromosome. See, these two are deleted in the chromosome. Let us take one example of this deletion. Naturally, any change will cause some abnormality, some disease. So, one example of a disease due to deletion is the Crideochart Syndrome, which is caused due to the deletion of the small arm. Deletion of small arm of chromosome number 5. So in this disease, the child, when it cries, Then the cry is like a sound of a cat. There are many more symptoms associated with the disease, but because of this sound, it is known as Crideochart. This is due to deletion. Now take this duplication. Duplication, कोई जीन 2 बार हो गया, ठीक है? Now this was a chromosome having the genes A, B, C, D, E, F, G, H. Let us take one homologous chromosome of it. I will show you the duplication in the homologous chromosome A, B, C, D, E, F, G and H. Now the normal chromosome, now this will undergo duplication. यही वाला पार्ट CD यहाँ से निकला और आके यहाँ पे जुड़ गया. So how it will be? This is A, B, C, D, then again C and D, C, D and E, F, G. So extra gene and extra products. This one, this duplication example, let us take. This is found in the chromosomes, the chromosome of salivary gland of Drosophila. ये हमने क्या देखा? ये हमने देखा कि एक क्रोमोसोम है, उस क्रोमोसोम का कोई पार्ट मिस हो जा रहा है. The genes are getting deleted or the gene becomes extra. There are many more such chromosomal aberrations. This one understood, chromosomal aberration. Now let us see what is point mutation. Point mutation. A very good example of it is sickle cell anemia. Sickle cell anemia. In this, the normal sequence of the nitrogen bases in DNA, it is GAG, and on another strand, on the template strand means the one which forms the mRNA. This is the complementary one. So this is C, T and C. And this is the DNA structure. Now the mRNA which is formed, it is complementary to this one, mRNA. So this is GAG, means the same thing. This is complementary to this one also. So you get the same. So this is GAG, the normal mRNA, which forms the amino acid glutamic acid at the sixth position of beta chain of hemoglobin. So this is the normal one. Normally ये ग्लूटामिक एसिड रहता है हीमोग्लोबिन के प्रोटीन चेन के 6th पोजीशन पे. नाउ द म्यूटेशन हैज ऑकर्ड. दिस इज पॉइंट म्यूटेशन. सिर्फ एक सिंगल पॉइंट पे म्यूटेशन होने वाला है. कहाँ पे? एट द सेंटर ना. दिस G and C remains the same. Now this A will become T. So this is G, and so this one will become C, A and C, and the mRNA, this mRNA, it will be GUG. GUG is the abnormal codon. Now it is going to place a wrong amino acid at 6th position. Now this glutamic acid, it is replaced by एक villain. that is valine. It is replaced by valine. And this causes such a serious disease called sickle cell anemia. Understood? Only at a point there is mutation, due to which there is change here at this one point, which has caused the disease sickle cell anemia. Done. Now we see what is frame shift mutation. Frame shift mutation, students. Frame shift mutation. In the DNA strand, the two DNA strands, the nitrogen bases, 4 types of nitrogen bases, A, T, C, G, they are arranged. Na? So if this is one DNA strand, this is another DNA strand. If this is A, T, let us write at the, no, we cannot write at the top. So if this is A, let us do this a little below. So A, T, G, A, C, G. The sequence can be any. The A, T, C, G are arranged there. But once this sequence is put, after that, the other one is always complementary. A with A, T pairs, then A, this is C, this is T, this is G, C and G, G and C. This is it. Now, the codon, the DNA strand, these two strands, out of the two strands, one of it will code for what? mRNA. So, the one which codes for mRNA, it is called template strand. So, if this is the template strand and it will form mRNA. तो ये जो mRNA बनाता है, mRNA is complementary to these bases. So, it will form A, U, G, A, C, G. ये बन गया आपका mRNA. So, there is a frame. This is a frame. You are reading. This is the reading frame. A, U, G, A, C, G. Then due to mutation, there is a shift in the reading frame, which totally disturbs the whole reading frame. तो अगर ये frame change हो जाए, then it will start coding for another amino acid, or it will not at all code for any amino acid. When there is deletion or insertion at a point of one or two nitrogen bases, then it disturbs the frame. But if it is 3 bases deleted or 3 bases inserted or multiple of 3, then again the reading frame becomes corrected. Why it happens so? This is only because this codon is a triplet one. ये तीनों मिला के बोलेंगे कि हाँ भैया ये एक अमीनो एसिड को हम यहाँ बैठने की परमिशन देते हैं. ये बोलेगा अच्छा मैं A2 को परमिशन देता हूँ कि फिर वो अमीनो प्रोटीन चेन में नेक्स्ट बैठे. So how this frame gets disturbed and why after when 3 are inserted or 3 are deleted then it becomes corrected, वो तो आपको समझ में आ गया. क्योंकि जैसे एक को हमने हटा दिया तो ये 2 बन गए, तो ये 2 बन गए, 2 नहीं रहने वाले, वो तुरंत यहाँ से सामने वाले से एक को खींच लेता है. So now it will form a different amino acid which will cause some kind of disease. Fine. So let us see by taking an example. We will try to understand this with the help of a sentence. A sentence in which every word has 3 letters. The 3 letters, they are synonymous of the 3 nitrogen bases. Let us see how insertion or deletion causes frame shift mutation with the help of this sentence. This side, let us take insertion, and here the deletion. The sentence is Ram has red cap. Ram has a red cap. Now, just before this red, let us insert B. So now this sentence will become Ram has, this B we will take, and this B now will take the R and E from the red. So now it becomes meaningless. B, R, E, then D, C, A and P. Fine? It is not producing the same, giving the same message that it is a red cap. Now the next one. After the B, we add I. So now this sentence becomes Ram has B, I, R, E, D, C and A, P. You are observing that from this point there is shift in the reading frame. Now the next one, after BI, here we add G. So the sentence now becomes Ram has big red cap. So this red and cap has appeared again when we have inserted these three letters. This is a proof also that the codons are triplet. 3 का है इसीलिए तो 3 डाला तो ये फिर से appear हो गए. So it is a proof, this thing is a proof that the codon is triplet. Also it is a proof that, ये दो याद रखिएगा बच्चों. Also it is a proof that there is no, means the codon is contiguous. Contiguous मतलब continuous, means there is no comma full stop in between. This is read as Ram has big red cap. The next letter which will be there, it will not be a full stop. It will again code for some amino acid. So we have seen about insertion. Now let us take the same sentence and let us see what happens due to deletion. So the sentence, the same sentence, that is Ram has red cap. अब हम लोग क्या करेंगे, इस red को उड़ा देते हैं ना. चलिए देखें. So in the first instance what we will do, we will remove the R. When we remove the R, then the reading frame will get disturbed from this point. So it is become Ram has EDCAP. Fine. Now we delete E. When we have deleted E, now it has become Ram has DCAP. Doesn't make any sense. Now remove this D. When you remove this D, then it has become Ram has cap. This is again become meaningful that Ram has cap. The cap here, it has reappeared. This is again the same thing, that it is proof that the codon is triplet and the codon is contiguous. But of course, this red, this specific information has been lost. The specific information which is lost, how important it is, accordingly there will be some disease, some abnormality like that. So this is how frame shift occurs. Clear? Now we will see all different types of mutations we have completed. Now just let us see some factors, the factors which cause mutation, जिनकी वजह से mutation होता है. So let us move to mutagens. They are called mutagens, mutation generating agents. They are mutagens. Let us see what are mutagens. Mutagens, mutation causing or mutation generating agents. Mutagens are mutation generating agents. They are of three types, students. First one, they are physical mutagens. These physical mutagens can be ionizing radiations like X-ray and gamma ray, X-ray and gamma ray, or non-ionizing radiations, non-ionizing radiations like UV ray. Then there are chemical mutagens. The chemical mutagens like the fumes of tobacco. Fumes of tobacco. Then there are biological mutagens also. Biological mutagens like in case of viruses, the viruses they have cancer causing genes. These genes of viruses they are called viral oncogenes. These viral oncogenes they cause mutation, and then you will be surprised to know that every individual of us, everyone is having some mutation causing genes called cellular oncogenes. Cellular oncogenes, but they are in inert forms. They are called C-onc. They are called C-onc, and if anytime they become active, then they cause alteration in DNA structure. So that was all, students. Hope you have understood mutation properly. Do watch Sunday videos. This every Sunday is devoted to concept building. So watch that on Sunday. And if you are new to my channel, then do subscribe, like and share. Thank you for watching the video.